So it's 8:05, so we'll start. I can admit people as and when they come in. Let me share my slide. Can someone tell me if I'm audible and if my slide is visible?
Yes.
Okay, thank you. So I'll start the session. Give me a second, please. So, good evening everyone. My name is Aishi and I am the PMRF TA for this course Applied Environmental Microbiology. I am also a PhD student, second year PhD student in the Department of Chemistry, IIT Bombay. And today is the fourth live session of the Applied Environmental Microbiology course. And we'll be discussing some of the sample problems. Hopefully these problems will help us discuss some of the core concepts and we'll be able to solve some assignments together. So we'll move on to the first question. Okay, where is it now? Yeah. So organomercury lyase makes the cell resistant to mercury poisoning. Organomercury plays a major role in bioaccumulation of mercury. MER gene is essential for formation of methyl mercury, and organomercury is more toxic than metallic mercury. Sorry, yes. I have already shown the answer. Sorry, this was not. Will anybody like to try? Which of the following statements is true? Anybody? I actually showed the answer before only. Okay, so these three statements are true. That organomercury lyase makes a cell resistant to mercury poisoning, and organomercury plays a major role in bioaccumulation of mercury, and organomercury is much more toxic than metallic mercury. So we'll learn a little bit about the topic over here. So as we know, mercury is a very highly toxic metal, and there are basic three forms of mercury that are very abundant in the atmosphere. That's elemental, inorganic ion, and organomercurial compounds. And the ability of bacteria to detoxify some of these mercurial compounds by reduction and volatilization is conferred by these MER genes, which are usually plasmid encoded, although chromosome resistant determinants have also been occasionally identified. So we all know that plasmids are basically extra chromosomal circular pieces of DNA that some bacteria have, and via these plasmids we can actually find the antibiotic resistance genes, and there are many kind of genes that are possible on these plasmids. Basically they are not a part of the chromosome, but they are extra chromosomal components of the cell, of the extra chromosomal DNA extra chromosomal genetic components. And this MER genes are also mostly present in the plasmids of this bacteria. So there's this protein encoded by MERB, organomercurial lyase, which is also known as alkyl mercury lyase, and this basically mediates the first of the two steps in the microbial detoxification of organomercurial salts, and the other is catalyzed by mercury reductase. And this organomercurial lyase catalyzes the protonolysis of this carbon and mercury bond in a wide range of organomercurial salts. These salts can be primary, secondary, tertiary, alkyl, depending on the positions and this conversion happens to mercury and the respective organic compound, and this mercury is subsequently detoxified by this mercury reductase. So as we know, organomercurials are very toxic because these can be very easily absorbed by inhalation or ingestion, and in ruminants methyl mercury methyl mercury is demethylated to inorganic mercury, decreasing this absorption. And dermal absorption, although slow, can also be sufficient to cause a toxicosis. And these absorbed organomercurials are then transported to RBCs, and then they get distributed all throughout the body, and these are very toxic because they can reach the brain by crossing the blood-brain barrier and placenta, and hence fetal blood levels can exceed maternal blood levels by up to 20%. And these organomercurials can also get secreted into the milk, and this biological half-life of organic mercury in humans is approximately 65 days. So that's why methyl mercury is the most toxic of the organic mercurials due to its persistence and how it can easily cross the blood-brain barrier and cause functional difficulties in the brain. And methyl mercury that is not sequestered within the nervous system is demethylated and primarily excreted, but mostly methyl mercury in the form of methyl mercurate is easily crossing the blood-brain barrier, and when something toxic reaches your brain, I think that's the end of the beginning. That's the beginning of the end, sorry. So that's why organic mercurials are much more toxic than any other form of mercury, because of this point that it crosses the blood-brain barrier very easily. Also, these genes, the MER genes, are the bacterial mercury resistant genes, and they are also they also play huge roles in the biogeochemical cycle of mercury. So as I said, methyl mercury, which is the most toxic form, it also gets biomagnified. This process is called biomagnification in the aquatic food webs. Like you all know that there's a food web, there's a food chain, and finally there's a food web depending on the production and the consumption, and this mercury is biomagnified in these aquatic food webs, and this causes toxicity to humans and animals basically that consume contaminated fish. The fish, if there are some fishes that have mercury sequestered inside their body, and when humans ultimately have these fishes, the mercury ultimately reaches their body, and since mercury is water soluble and it absorbs strongly into iron oxides, it interacts with other dissolved organic matter also. But mercury with the mercury zero, this is the least toxic form, and can be present both as liquid and gas, and can evaporate from surface waters and soils. So all the this MER encoding genes, these are formed in the MER operons, which converts this mercury 2 to mercury 0, which is the least toxic form, and this confers some kind of resistance. So these are some broad spectrum MER operons, and they contain some additional gene encoding this MERB, or which is also known as alkyl mercury lyase, and this degrades alkyl mercury 2 and methane. And there are other MER operons that contain genes for transcriptional regulators like MER-R and MER-D. So regulation of these MER genes and the enzymatic activity of MERA is known in great detail for some operons and enzymes. And so this we have already discussed about the inorganic mercury salt and how it conjugates with cysteine and gets absorbed well and can easily be transported to the it can enter the cell where mercury easily detaches. It can traverse placenta. That's why fetal may have 20% more mercury concentration than adults, and then it can enter brain oxidized to elementary mercury, and that is retained in brain because it cannot pass through the blood-brain barrier. So the elementary mercury cannot cross the blood-brain barrier back, and that's why it gets retained in the brain, making it all the more toxic. So that's why organic mercury can cause severe brain damage in addition to some other organs. So although both inorganic and organic mercury compounds are in fact toxic, it is the source or the pathway of exposure that is an issue. So how the exposure happens by eating mercury contaminated fish, or how it can go into the blood-brain barrier and can transform into elemental mercury and not cross the blood-brain barrier back, that is what causes this neuronal apoptosis and can cause severe brain damage. So that is why it is written that this is most prevalent via the pathway of food consumption. Like if there's as we talked about biomagnification via biomagnification in the food, aquatic food webs, that's what happens mostly. And organic mercury compounds are also highly lipophilic and then tend to accumulate within the fish tissues, and after consumption these compounds in turn can bioaccumulate in myelin, these fatty livers that coat and protect nerve fibers. The myelin sheath, there is a myelin sheath on the neuron, the axon part of the neuron, so these can easily bioaccumulate in the myelin sheath and the spinal cord of the central nervous system. So that's why mercuries are like really toxic. So it poses basically two dangers. It can be vaporized, and mercury can easily cross the brain, and it is biotransformed into organic mercury by bacteria at the bottom of the legs. So this is what happened to this process led to the Japanese tragedy at the Minamata Bay in the late 1950s, and over 800 people were poisoned. And inorganic mercury salts are also toxic because they are water soluble. They can irritate the gut and cause severe kidney damage. But organic mercury compounds are much more toxic because of this biomagnification issue, because they are fat soluble, because they can cross the blood-brain barrier, can deposit on the myelin sheath of the neurons and can cause severe brain damage and can cause severe spinal cord damage as well. Basically, the central nervous system can be easily damaged, and this can ultimately lead to neurological damage. That's what we have been talking about. So that was that. So moving on to the next question, which of the choose the following, choose the relevant option. Biofilms comprise of the best possible combination of cooperation and competition, and population is a collection of multiple microbial communities. Which one do you think is the correct answer? Anybody would like to try? Okay, so only one is true, that is biofilms comprise of the best possible combination of cooperation and competition, and that is because, as we have already talked a little bit about biofilm, what happens when biofilm formation occurs. Bacteria basically stays in two stages. One stage is from the sessile bacteria, which attaches easily, and the other is the phytoplankton stage. But in the sessile stage, when bacteria is attaching, it reversibly or irreversibly attaches to a solid support, and that's when they become irreversibly attached to a surface and they form a matrix, as we can see from the sketch over here. And quorum sensing happens. Quorum sensing is basically how a lot of bacteria they behave like social predators. They can come together in bunch, and they kind of secrete molecules, and that helps them communicate amongst each other. So quorum sensing, this begins and the cells become layered on top of each other. That's called the maturation phase, and then 3D structured colonies are developed, and that reaches the maximum thickness. And dispersion of this planktonic bacteria from the matrix is known as dispersion. That's called dispersion. So as we can see here, motility and less active movement to attach site cell surface appendages enable the cell to overcome repulsive forces associated with cell surface. Then cell adsorption happens. There is passive attachment with weak bond van der Waals, and over time there is some irreversible attachment with strong stable bonds occur, like ionic, then covalent, then dipole-dipole interaction. There's a cell-to-cell signaling, or the quorum sensing starts. There's onset of EPS production, or the extra polymeric production. There's development of this biofilm structure. Then that leads to the maturation phase, where there is convective and diffusive transit of O2 and nutrient. There's replication growth. There's secretion of EPS, and then there's dispersal and detachment. These are the basic steps of a biofilm formation. And these are some confocal. The B, C, and D show some epifluorescence microscopy view of biofilm that were grown on a stainless steel surface, and they have been stained with DAPI. And this biofilm formation of E. coli, the C represents some biofilm formation of E. coli and titanium oxide, and it has been seen through scanning electron microscope. This D image shows a confocal laser scanning microscopy image of a biofilm formation. Now we all know that there are some kind of social interactions that are happening in biofilms, and these can be most broadly categorized into communication, cooperation, and competition. So as we know, bacteria often exist in these biofilms, which are surface adhering or free floating groups of cells that are bound together by a secreted polymer matrix. And these microbial collectives are important for bacterial occupation of diverse ecological niches. They can not only contribute to biogeochemical cycling, and they can also cause some kind of disease in multicellular organisms. And within biofilms, bacteria interact with each other closely through cooperative phenotypes, such as the production of digestive enzymes, and some kind of antagonistic phenotypes, such as the expression of type V or type 6 secretion systems. So the evolutionary dynamics of these social phenotypes of interaction depend on their cause and the effects on the other cells. So many bacterial social phenotypes involve the secretion of products that affect neighbors in a distance-dependent manner. As a result, interaction networks within biofilms are largely determined by the spatial structure of the biofilms, that is, the rearrangement in the space of different clones, strains, and species. So when biofilms are segregated into clonal clusters, the neighborhood of a given cell mostly contains clone mates, and natural selection often favors the secretion of compounds that benefit all these recipient cells. So, however, when different strains and species are spatially mixed within biofilms, cells primarily interact with each other with other genotypes, and antagonistic behavior is often favored. So, when there are different strains and species also coming in contact, that's when mostly antagonizing behavior is seen. And under certain circumstances, between species, commensalism or mutualism can also evolve and remain stable. So we know mutualism is that when there is a kind of an interaction between two species where both of them benefit from each other. That's called mutualism. Commensalism, I think one species benefits and the other doesn't have, there's no benefit to the other, but also there's no harm to the other. And when there's in the situation where one species benefits and the other species is harmed, that is called parasitism. So in certain cases, when different strains and species appear in this biofilm, under certain circumstances, commensalism or mutualism can also occur. So basically, cooperative and antagonistic phenotypes fall under the control of sophisticated sensory mechanisms such as competition sensing and quorum sensor that are evolved to help account for the variation in exposure to other strains and species in space and time. So what happens, these regulatory mechanisms, these help to reduce the marginal costs of social phenotypes, and they can maximize their fitness, impacts, and ensure that the correct recipient cells are targeted. So both cooperative and antagonistic behaviors feed back onto the population spatial structure by locally altering the growth rates of other cells and thus changing the local biofilm composition. And we know that many bacteria and unicellular eukaryotes have evolved strategies for actively altering this biofilm population structure, like either through selective adhesion that spatially assorts the biofilm into groups that contain one or more specific genotypes, or through the secretion of extracellular matrix components that spatially organize these biofilm dwelling cells. And this coordinated division of labor and complex network between close proximity cells in the biofilm matrix, this is what encourages socio-microbiology. And the important features of social interactions in biofilms is intercellular signaling and metabolic activity. So when there is a high species diversity and cell density, the exchange of metabolites between species is easy in a biofilm. And intercellular signaling and the exchange of metabolic byproducts are not present in planktonic cells which are not in a attached biofilm kind of structure. So cooperation or competition between the cells of biofilm develop the socio interactions in biofilms. There can be chemical communication or electrical communication that might be involved in this kind of cooperation, and moreover social interactions are also involved in cooperative metabolisms such as nitrification process, in which ammonia is converted to nitrite by ammonia oxidizing bacteria in the absence of UDAs. Then there is nitrite oxidizing bacteria that can convert nitrite into ammonia by using enzyme UDAs. Then the metabolites of ammonia and nitrite oxidizing bacteria are exchanged with each other to reduce loss and increase the effective substrate uses. So then there's an example of cooperation in biofilms, which is the synergistic degradation of linuron. It's a toxic herbicide found in some mixed species biofilms such as Hyphomicrobium sulfonivorans or Comamonas testosterone, and there's another Variovorax or something like that, basically mixed species biofilms. And all these biofilms will come together, there is a synergistic degradation of a toxic herbicide called as linuron. And then there's some mixed species biofilms that show more tolerance than single species biofilms for this linuron tolerance. So also synergistic interaction is also observed in many biofilm forming consortia. It can be biofilm together with some other organism like fungi or cyanobacteria on buildings and rocks. Then there can be negative interactions, for example competition can also be present in biofilms. So the production of any kind of antibiotic or bacteriocins or extracellular membrane vesicles containing enzymes that might reduce the growth or kill competing organisms are mechanisms, biofilm cells that create competition. So some species of the biofilm might be producing some kind of special antibiotics, or can be secreting some kind of special enzymes or substances that might kill off the other species in the mixed biofilm structure. So this is what creates a kind of competition. As you can see from here, this inhibits quorum sensing and can form a competition over here. However, nutrient reduction and the inhibition of autoinducers is also a type of killing strategy that is presented by bacteria. So when the nutrient quantity reduces, or there's some kind of autoinducer inhibition that happens, that's also is kind of a competition that is taken up by some bacteria in this biofilm. ecosystem basically. So in this biofilm, when there's a mixed species biofilm, there's chances of cooperation, competition. Communication we all know can be done by quorum sensing and all that, but then cooperation and competition can also occur. So that are some kind of these are some kind of the social interactions that happen in biofilms. So the first option is true. Biofilms comprise of the best possible combination of cooperation and competition. True. Population is a collection of multiple microbial communities. So the reason I thought this is not kind of true is that when we talk about evolution, we say that evolution can be defined as change in the genetic makeup of populations, and being defined as a locally coexisting representative of species. Populations are basically the units of diversity that are used when we wish to measure some kind of species diversity, basically to assess ecological interactions as well as ecosystem stability and resilience. For microbes, however, populations have been very difficult to define, and we use arbitrary units of diversity to measure the genetic makeup of these communities. So this difficulty in defining populations is of course rooted in the absence of a biologically meaningful species concept for bacteria and archaea. So without clearly defined populations, many of the most fundamental questions in community ecology are difficult to answer. For example, do disturbances lead to changes in genotypic composition within populations or to species turnover? So differentiating between these possibilities is a meaningful question, because shifts in genotype within a population may be far less disruptive to ecological networks than wholesale changes in the species composition. So this question is basically at the heart of understanding the key dynamics between microbial communities, including the human microbiome. So that's why defining bacterial and archaeal populations, and by extension species, is definitely an important endeavor. But can we do it? Is microbial diversity organized into natural units to which we can actually ascribe biologically meaningful properties? So specifically, two fundamental evolutionary processes organize coexisting genotypes into units through which adaptations can specifically spread, giving rise to ecological units with clearly different dynamics. So if we can actually define microbial populations in such a way, then we may be able to apply the rich evolutionary and ecological theory developed for animals and plant populations. If not, then we might need a fundamentally different theory and approach. So that's why we know that option 1 is definitely true. Option 2 might be a little dicey. So moving on to the next question. Ecosystem combines biotic and abiotic factors of the environment. What do you think? Anybody? Sorry? Did you say something? Did you say true? I am not able to hear you properly, your voice is tracking, but okay, yeah, it's true. So ecosystem combines both the biotic and abiotic factors of the environment. So if we can see this rough sketch, so the factors of ecosystems, there will be abiotic components such as the inorganic components like oxygen, nitrogen, organic components such as carbohydrates, proteins, fats, basically structures made of the organic structures. Then we have the physical components such as sunlight and soil, which are also very indispensable for growth and nutrients. And the biotic factors of the ecosystem will obviously have the living entities of the ecosystem, like the autotrophs or the producers, the heterotrophs. Even amongst the heterotrophs, we have the consumers. Then we have the primary, secondary, or tertiary, how a food chain is formed. The primary consumer, grasshoppers. Who eats the grasshopper? A frog can eat the grasshopper. So that's the secondary consumer. And then we have the tertiary consumer, which is a snake. A snake can eat the frog as frog. So that's how the consumer food chain is formed. And amongst the heterotroph, there is another category that we can talk about called the decomposer. So these are basically the two factors of ecosystems. They can be abiotic factors, and then they can be biotic factors of the ecosystem as well. Next question. ORP determines the functional class of microbes in an environment. True or false? Anybody? ORP is oxidation reduction potential. Anybody would like to try? So ORP is basically oxidation reduction potential. So what is ORP? It basically measures the ability of water body, suppose a lake or a river, to cleanse itself or to break down its waste products such as contaminants and dead plants and animals. So when the ORP value is high, there's lots of oxygen present in the water. This means that bacteria that decompose dead tissue and contaminants can work more efficiently. In general, the higher the ORP value, the healthier the lake or river is. However, in healthy lakes or rivers, there's less oxygen and therefore lower ORP values. So as you get closer to the bottom sediments, this is because there are many bacteria working hard in the sediments to decompose their tissue, and they use a lot of available oxygen. In fact, oxygen disappears very quickly in the bottom centimeter or two, and thus ORP falls quickly. So it's basically measured in addition to dissolved oxygen because it can provide scientists with additional information of the water quality and the degree of pollution if present. Also, there are other elements that can function like oxygen and contribute to increased ORP. But why does oxidation reduction potential matter? So this ORP depends on the amount of dissolved oxygen that is in the water as well as the amount of other elements that function similarly to oxygen. So though not technically correct, oxygen and other elements that contribute to high ORP effectively help decompose or eat things that we don't really want in water, such as contaminants and dead tissues. So when dissolved oxygen is low, toxicity of certain metals and contaminants can increase, and there's lots of dead and decaying material in the water that cannot be cleared or decomposed, and this is obviously not a healthy environment for fish or bugs or any kind of aquatic life. In healthy waters, it doesn't happen. ORP should read high. So we might expect low ORP in waters that receive sewage inputs or industrial waste. But how is this ORP or the oxidation reduction potential measured? So it is measured directly in the water body that we are investigating using a kind of ORP sensor. It is measured in millivolts, and the more oxygen that is present in the water, the higher the reading is. It can be either above 0 or below 0. So it's nothing but a numerical index of the intensity of oxidizing or reducing conditions within a system. And water samples can contain different types of oxidizing and reducing substances. Because the ion species cannot be identified by the ORP value, the measurement of ORP in natural water is in millivolts rather than any kind of concentration units. So positive values for ORP basically indicate oxidizing conditions, whereas negative values indicate negative, like reducing conditions. So it's a quantifiable measurement of the tendency of a molecule or ion to gain or lose electrons basically, and this term is also used to basically monitor drinking water and wastewater treatment processes for oxidation state due to the addition of halogenated disinfectants such as chlorine. So these and some studies have already shown a strong correlation of this oxidation reduction potential and the bactericidal activity of chlorine. So this ORP is also an indicator of the level of disinfectant in the wastewater effluent, and the presence or absence of chlorine will result in a positive ORP value, and the presence of substances like hydrogen sulfide will result in a negative ORP value. And we all know that redox potential and pH, these are very inherent or intrinsic factors of food habitat, and basically reduction of oxidation potential describes the differences in electrical units in volts or millivolts, where one system is oxidized and the other system is reduced, and during this process a reduced substance loses electrons and is oxidized to an oxidized substance that is subsequently reduced. So in biological systems, oxidation of substances is a basic principle of energy generation. So energy rich compounds are oxidized stepwise in cellular catabolism, and in the presence of sufficient oxygen, most microorganisms perform classical oxidative phosphorylation. And electrons from various oxidized electron donors are transported via electron transport chain to the electron accepted oxygen, which is reduced to water, and this process is further coupled to proton translocation via the cytoplasmic membrane in bacteria or the inner mitochondrial membrane in the fungi. We have talked about this in the last class. And the electrochemical pH gradient that is formed is basically used to drive the ATPase to form ATP from ADP and PI. So changes in the environment oxygen tension have a drastic impact on the catabolic functions. And so in the absence of oxygen, what happens? It is possible for bacterial and some fungal cells to achieve anaerobic ATP formation in two ways. Oxygen can be replaced by alternative electron donors, electron acceptors, such as nitrate, fumarate, dimethyl sulfoxide, then thiosulfate, to sustain further electron transport and membrane potential driven ATP synthesis. And in biological systems, electrons are carried from the electron donor with more negative or less positive redox potential stepwise to acceptors, which have less negative or more positive redox potential. And this covered redox potential differences combined with proton gradient formation determine the rate of energy conversion. So the larger the differences in redox potential between the electron donor and acceptor, the better the chances of efficient energy conservation. This explains why oxygen is still the most valuable electron acceptor in nature, basically. So, and the second possibility that can happen in the absence of oxygen is the level of substrate degradation, or it's called fermentation. So in the absence of electron transport chains for energy generation and NAD plus regeneration, fermentation processes generally occur, which only partially oxidize the substrate, leading to the excretion of energy rich metabolic intermediates and overall poor energy conservation. So on the basis of their growth capabilities in the presence and absence of free oxygen, microorganisms can henceforth be grouped as aerobes, anaerobes, and facultative anaerobes and microaerophiles. So strict aerobes, strict aerobes or obligate aerobes require oxygen as electron acceptor for energy generation. So you can see the obligate aerobes basically bunch up at the top of the media so that they can get enough oxygen. They can be exposed to enough oxygen. But strict or obligate anaerobes cannot grow in the presence of even small amount of oxygen. That becomes toxic for them due to the lack of this superoxide dismutase activity, which is necessary for this oxygen radical scavenging. And strict anaerobic microorganisms mostly grow on the basis of fermentation process. Then there are facultative anaerobes, which use oxygen if available and grow anaerobically using alternative electron acceptors. Then we have the microaerophiles, and the microaerophiles grow best at low oxygen tension. So they require oxygen but are not in need of the oxygen as such. So they grow closer to the exposed surface but not exactly at the exposed surface. So clearly the presence or absence of oxygen, and as a consequence the changing redox potential, determine the mode of energy generation in different environments, including food, and the pH determines the concentration of hydrogen ions in a system. So that is how, and the pH of the environment basically has a profound effect on the growth and survival of all these microorganisms. Like changes in the external pH can directly influence the status of the proton gradient and the cytoplasmic membrane of bacteria, and this is responsible for ATP synthesis, transport processes, and motility. So since many microbial species require homeostasis of the internal pH for growth, changes in the external pH induce complex adaptation processes. So in order to adapt to these prevalent living conditions efficiently, the cells must first detect this environmental stimuli and in turn respond with changes in their metabolism. So this control is mostly due to transcriptional regulation of the genes that encode the individual proteins involved in the biochemical function specific to certain environmental conditions. And which is why this is a very important consideration, and this potential is very important for biology of microbes, and certain organisms can only survive in certain environments with respect to their redox potential. So this sort of creates this cyclic relationship between microbes and the environment. So when we think of the environment influencing which organisms can grow, we also have to note that microbes are influencing their own redox around them. So they're essentially manipulating the environment via metabolism. So to put this entire thing in context, microbes, while the environment determines which microbes can grow, microorganisms can also strip oxygen from the environment and produce reductive metabolites and basically manipulate the environment and subsequently the kind of microorganisms that will grow in that environment. So that is why this happens. That is why this ORP is very intimately connected to the kind of microbe interaction that happens in the atmosphere. So with this oxidation reduction potential, it can be used to classify microorganisms. So this is the most important slide related to this question. So we want to classify bacteria based on oxygen requirement. This is what we do. It can be classified basically widely into 3 categories depending on their action or towards free oxygen, or more precisely oxidation reduction potential, or ORP. So as I already said, obligate aerobes require free dissolved oxygen to oxidize organic matter and to live and multiply. So these conditions are required as aerobic process, while as anaerobic bacteria don't really require oxygen to grow. They can grow in the absence of oxygen. They can take like they can use inorganic salts, or they can use other electron acceptors, and this mode of operation is called anaerobic process. Facultative bacteria can use both free dissolved oxygen which are available, but can also multiply and respire in the absence. So E. coli is a facultative bacteria. It can grow in the presence of oxygen, but it's oxygen, it can grow in the presence of oxygen, but even without oxygen it can show growth. So this is how classification of bacteria is possible based on ORP, or the oxidation reduction potential. Moving on to the next question. Microbial conversion of sulfur to hydrogen sulfide is termed as desulfurization. Anybody? Anybody would like to try? Does anybody know what desulfurization is? Okay. So it's false, but we'll talk a little bit about the sulfur cycle. So sulfur, as we know, is one of the most versatile elements in life due to its reactivity in different reduction and oxidation states, and it is the element, I think, with the maximum number of highest number of allotropes, but only a few are actually found in nature and occur in biological systems. So the inorganic sulfur compounds of biological relevance which occur in the biological sulfur cycle are elemental sulfur, sulfate, sulfite, thiosulfate, polythionates, sulfide, polysulfides, all these. So sulfur, as you can see from this, sulfur can actually adopt a variety of oxidation states ranging from 2 to +6. And inorganic sulfur compounds of intermediate oxidation states can serve as electron acceptors or donors in redox processes. So in contrast, sulfate and sulfite cannot be further oxidized or reduced respectively, and they are therefore the final products of most sulfur oxidation and reduction pathways. So the biological roles of inorganic sulfur compounds are rather restricted. Either they serve as acceptors or donors of electrons for dissimilatory energy generating electron transport, almost exclusively among prokaryotes, or they are employed as sources for sulfur assimilation, very common in prokaryotes as well as in algae, fungi and plants. So despite its toxicity, like it's 5-fold more toxic than carbon monoxide, hydrogen sulfide, or H2S, is still a fundamental molecule in both anaerobic and aerobic organisms. And like most studies about hydrogen sulfide have been devoted to its toxic effects with little attention to its physiological function. So the liberation or the release of H2S is controlled not only by the rate of its production by sulfate and sulfur reducing prokaryotes, but also by its tendency to rapidly precipitate as metal sulfides, its pH dependent speciation, and its fast biological and biological and chemical oxidation. Only the protonated compound hydrogen sulfide is volatile, whereas sulfide or S2 dominates under alkaline conditions, and at neutral pH most of these inorganic sulfides are present as bisulfide and anion. So this is what happens in sulfur cycle. Sulfur can be present in many amino acids as well, and the sulfur cycle involves oxidation reduction between all these sulfur forms. So the basic steps involved in the sulfur cycle are these: sulfur or sulfide oxidation, sulfate reduction, sulfur reduction, organic sulfur compound oxidation or reduction, and desulfurylation. So what happens in sulfide or sulfur oxidation? It's carried out by prokaryotes under aerobic and anaerobic conditions. And under aerobic conditions, what happens? H2S is spontaneously oxidized at neutral pH to elemental sulfur. And elemental sulfur is oxidized to sulfates by certain chemolithotropic bacteria such as Thiobacillus, I think also Thioplaca, then Veggiatoa. And if light is available, then H2S can be used as electron donor to carry out photosynthesis under anoxic conditions by phototrophic sulfur bacteria like Chromatium and Chlorobium. So this is what sulfide and sulfur oxidation means. Sulfate reduction is that when sulfate is present abundantly in the habitats, different groups of microorganisms are capable of carrying out this sulfate reduction. So there was a scientist who described the use of sulfate as a terminal electron acceptor during anaerobic respiration to form H2S or hydrogen sulfide, and this process is called dissimilatory sulfate reduction or DSR. And the anaerobic bacteria capable of carrying out DSR are Desulfovibrio, Desulfococcus, Desulfotomaculum, and this reaction by sulfate reducers requires organic carbon sources like pyruvate or lactate. And H2S, when accumulated in such habitats by the action of sulfate reducers, is very toxic to the aerobic organisms requiring oxygen to survive. And the reduction of sulfate to H2S for building up amino acids and proteins is called assimilatory sulfate reduction. So this H2S that is produced is immediately assimilated or incorporated into organic compounds. Then we move ahead to sulfur reduction. So the dissimilative sulfur reducing bacteria can reduce elemental sulfur to hydrogen sulfide. A prominent example is disulfurmonas, an obligate anaerobe. And under aerobic conditions, organisms such as Pseudomonas, Protease, and Salmonella are also capable of performing this reaction. So this is the biological sulfur cycle, and all the bacteria and their roles have been identified. So these solid lines basically indicate aerobic reactions, the dashed lines indicate anaerobic reactions, and the dotted lines, they indicate basically both aerobic and anaerobic activity. So desulfurylation by many aerobic and anaerobic prokaryotes is happening over here, and assimilatory sulfate reduction by many aerobic and anaerobic microorganisms, then dissimilatory sulfate reduction, all of these are happening here, and the disproportion of thiosulfate by disulfobrio and disulfobrio and disulfocapsa, these are also happening in this step. So sulfide and sulfur oxidation by colorless sulfur bacteria is happening in steps 1 and 2. Step 3 is showing, where is step 3? Step 3 is showing sulfur reduction by the anaerobic microorganisms. Step 4 and 5 show anaerobic sulfide and sulfur oxidation by purple sulfur bacteria and green sulfur bacteria. And step 6 is basically showing the sulfide reducing bacteria. Moving on to the next question. Microbial communities may undergo succession due to evolution. Anybody? For this, we need to understand what succession and evolution mean and what is the primary difference between these two terms. So we discussed succession in the last class. Anybody would like to try this question? Just anything is fine. Don't worry about being right. Just let me know if you have any opinion. We'll discuss.
True.
You think it's true? Okay. So do you know the difference between evolution and succession? Okay. So I'll tell you why I think it's false. So basically we need to learn the difference between succession and evolution. So as we know, evolution occurs inside the population of animals, bringing about speciation. So it doesn't happen overnight. Over the years, over millions of years, some parts of the body change. Some things happen due to the environment or due to the competition, interaction with other species and everything. And this basically brings about changes in the population of animals, bringing about speciation, different species formation. However, succession occurs with only one population, however an entire environmental network. So this implies that after some time, as advancement happens, the natural progression will result. And environmental progression can't occur without evolution. However, development can occur without natural progression. For instance, after some time, we can watch the finches on the Galapagos Islands advancing and turning out to be new species. This is simple evolution. I think the Galapagos Islands, the finches of the Galapagos Islands were a very important topic taken up by Darwin in his theory of evolution. So this is simple evolution. They are turning out to be new species. But over some stretch of time, in light of new types of finches, new bugs and new plants likewise evolve on this island. That is ecological succession. Succession, primarily succession is of two types, primary and secondary succession. So in first kind of succession, everything is starting from scratch when there's nothing on a ground before or any kind of rock before, and everything, first lichens arrive, then there are different kinds of small shrubs that are arriving, then there are trees, and finally the entire ecosystem reaches a climax community, a mature forest. This is primary succession, when there was nothing present there and everything is starting from scratch. So these are explained in the previous class. So you can go through the YouTube videos. I've already uploaded the previous class YouTube video, and I've also uploaded the presentation of the last class. So the diagram, everything was explained with diagrams over there, so that will be easier to understand. However, secondary succession happens when there was already a climax community present, a mature ecosystem already in place, but then there's some kind of natural disasters like wildfire happening on. It starts with one tree and then spreads easily throughout the entire forest. This is a natural disaster. It happens and it actually wipes out the entire living ecosystem over there, and then everything has to start over again. That is what we call secondary succession. So the gradual process by which ecosystems develop or change over a period of time is called ecological succession. Evolution, which is basically natural selection, is a mechanism of evolution, and in this case species adapt to the changing environment by bringing adaptation to the survival patterns and reproductive structures. So this natural selection or evolution and ecological succession, these are two different theories that work into different directions and fulfill different purposes. While one explains the process of evolution, natural selection, the other explains the stabilization of a community over on our planet. So in ecological succession, the populations that make up a community change, because but the characteristics of the individuals within the population will not change over time. So the populations change because we are starting with, in case of primary succession, we are starting on a new land. So we are starting with lichens and small shrubs, and then we move on to the bigger trees, then a mature forest, and the climax community. So this happens in ecological succession. The populations that make up this entire community, they change, but the characteristics of each of these individuals within the population, they do not change. Lichens will not start showing some different characteristics, or shrubs or trees or mature trees, these kind of trees won't, the climax community trees, they won't start changing their characteristics over time. But in case of evolution, what happens over millions of years, there's new speciation occurring. So new species are coming out of changes that have already been happening in previous species. There's natural selection. Natural selection will wipe out the natural selection, and then the survival of the fittest phenomenon. So there will be competition. They'll wipe out the least fit organism. They'll wipe out the least fit species. Then they'll like allow the fittest to survive. So ecological succession is basically something that happens to communities, while evolution happens to populations. So that these are basically two different parallel concepts, and they have two different, completely different purposes or goals. So if we have to compare what these two mean, I have tried to put together a difference table that I found on the internet. So ecological succession basically means the process of evolving a biological community over time. So there's a community that is started with lichens, it's followed by shrubs, smaller plants, smaller herbs, shrubs, and then it is moving on to trees. Natural selection or evolution, basically, this process favors the survival of organisms that are better adapted to the environment. So there are different kinds of organisms present in the environment. With time, with decades and centuries, millions of years, the organisms that are more favorable to survive in the environment, which are fit, which can actually adapt to their surroundings easily, will survive, and the least fit, which will not be able to survive, which will be vulnerable to any kind of change in the environment, they'll eventually die out. So that's what we call natural selection. Then ecological succession, this theory was given by scientists such as H.C. Cowles, Gleason, Clements, Robert Whittaker, John Curtis. However, the natural selection of the natural selection was proposed by Charles Darwin. We have another Lamarck's theory, which is also a very famous theory in terms of evolution. In ecological succession, this change that is observed, it happens within the community. In a community, some kinds of organisms, some kinds of trees were already present. They are followed by some other types of trees. But in natural selection does not directly affect the community. The change is basically observed within the population. But in ecological succession, the individual populations of the community do not undergo any change. So in the entire community, some things appear and some things disappear. Some things appear with time, but the individual population of the individual species, they do not undergo any kind of change. Then ecological succession, it is a gradual process that results in the development of a stable climax community from bare land and area. As I discussed in the last class also, there is a bare land or area, and this is gradual process that leads to the development of a proper stable climax community or a mature forest kind of space. In natural selection, it's a selection process basically that favors the survival and reproduction of the organisms that are suitable for the environment, that can get better adapted to the changes in the environment. In ecological succession, the simple organisms are replaced or evolved to higher organisms. That means at first there were simple organisms like lichen, as I described, and these are ultimately replaced by huge trees, which is basically the climax community. But in case of natural selection, it's a selection process. Basically, it's a choice. It's how the environment chooses the most fit of all the organisms. So this selection process favors the survival and reproduction of the organisms that are suitable to the environment. Anything else will get wiped out in the competition. And while the non-selected organisms are eliminated from the population. Then, in case of ecological succession, it provides the framework for establishment of a community. But natural selection is one of the major forces of evolution on our planet. In ecological succession, what happens? There's change in the climate, there's change in the soil, biotic changes also happen, and the desire to achieve stability are the main causes of ecological succession. As I said, in case of secondary succession, there probably was a very huge natural disaster like wildfire. All the trees caught fire and everything got demolished. So there was climate change, there will be soil changes, but with time there will be new organisms that will come back on those lands. There will be climate change, soil change, so the entire community will change with time, and this will ultimately reach a stable climax feature, which is called the ecological succession of the climax community. But in case of natural selection, it will occur in population, and 4 conditions need to be fulfilled, like reproduction, heredity, variation in physical characteristics and fitness, and high rates of population growth. So since this change happens in the population level, these are the 4 conditions that need to be fulfilled. The ecological succession are of 2 types, primary and secondary succession. The diagrams and the explanations are in the last class. You can check out the YouTube video for that. So primary succession and secondary successions are the 2 major types of ecological succession. However, natural selection can be directional, in a direction, stabilizing, and disruptive selection. These are some of the these are the categories of natural selection. Then ecological succession is important for the growth and development of an ecosystem by recognizing the older areas or destroyed areas, or initiating colonization on new areas. As new areas, matlab primary succession. By older destroyed areas means secondary succession. In case of natural selection, it's important for the process of evolution, as it can lead to speciation. Basically, the formation of the emergence of new species, the elimination of the less favorable traits, and the passing of the advanced and favorable alleles to the offsprings. So whoever will be more fit will survive. Whoever will be able to give their basically further their genes through reproduction will be able to survive. They'll pass off the favorable alleles of each gene to the offsprings. So for example, they have shown succession on land after volcanic eruptions. Secondary succession. This is a very good example of ecological succession. However, natural selection is a skeletal adaptation of the giraffe. So it was said before that giraffe neck used to be really normal, in the normal size of any 4-legged animal. But with time, we see that the giraffe's neck has it's huge now, right? So that's because at some point of time there was a lot of trees that the giraffe needed to reach. And for that they needed their height to be really proper, and with time, for decades and millions of years, stretching their neck to eat the leaves from really high trees made them evolve into the giraffe that we see today, with very huge and long necks. So this is a very prominent example that Darwin had talked about, not the Lamarck had talked about. I think giraffe is Lamarck's theory, talked about in the case of evolution. So moving on to the next question. All ferric reducing bacteria form one guild. Anybody? Anybody would like to answer?
True.
True. Okay. Yes, this is true. So before we talk about the question, we'll talk about what a guild is. So guild is basically any group of species that exploit the same resources, or that exploit different resources in related ways. And it's not necessary that the species within a guild occupy the same or similar ecological niches. So, and guilds are defined according to the locations, their attributes or activities of the component species. For example, the mode of acquiring nutrients, the mobility, the habitat zones that the species occupy or exploit can be used to define a guild. And the number of guilds occupying it is termed as disparity. So that's what basically guild's definition is. And members of a guild within a given ecosystem could be competing for resources such as space or light, while cooperating in resisting wind stresses, attracting pollinators, detecting predators, such as what happens in the case of savanna dwelling antelope and zebra. So a guild does not typically have very strict or clearly well defined boundaries, nor is it very easy to taxonomically rank or taxonomically define. A broadly defined guild will almost always have the constituent guilds. For example, grazing guilds will have some species that concentrate on coarse, plentiful forage, while others concentrate on low-growing, finer plants. Both are grazing, both belong to the grazing fields, but some of them focus on coarse, plentiful forage, and other focus on finer plants. And each of these two subguilds may be regarded as guilds in appropriate context, and each of those subguilds can also be termed as individual guilds, separate guilds, when in the right context basically, and they might in turn have further subguilds in more closely selective contexts. So basically context is what defines a guild. So this concept arises in several related contexts such as metabolic theory of ecology, scaling pattern of occupancy, and the spatial analysis in ecology. All of these are very functional and fundamental concepts of defining guilds. It is not similar in meaning to a taxoncene. A taxoncene is basically a group of phylogenetically related organisms in a community that do not necessarily share the same or similar niches. So nor is a guild the same as trophic species. Organisms are the same species that have mutual predators and prey. So that's how a guild is described. Basically, it exploits the same resources. It can exploit different resources in the same way. And with respect to context, a guild can have further subguilds, which can also be termed as separate guilds when the context is relevant. So this is how ecologists basically use a hierarchical classification system to describe organisms and their communities, and individual microorganisms that are genetically related and perform the same function function in an approximate location are referred to collectively as a population. Basically, populations that compete for the same resources are grouped into a guild, and all of the guilds in those populations that present in a specific environment constitute the microbial community. So these are basically this diagram specifies the complexity of interactions and how ecologists have been using a hierarchical classification system to describe organisms. So yes, all the ferric reducing bacteria, they are basically exploiting the same resources. So they can be it can be said that they form one proper guild. So it's true. She's correct. So moving on to the next question. Biofilms can give protection to the microbes in a harsh environment. True or false? Anybody? Do try. It's fine if I want to know your opinion. If you are trying to think in a particular way, and what do you think the answer might be?
True.
So yes, it's true. So these are some of the features of a biofilm. So we all know that biofilms are matrix enclosed microbial aggregations basically that adhere to biological or non-biological surfaces, and they represent a significant and incompletely understood mode of growth for bacteria. It's not properly understood yet, and biofilm formation appears it appears I think in the fossil record also, like even 3.25 billion years ago there were biofilm formations, and is common throughout a diverse range of organisms in both the archaea and the bacteria lineage, including the living fossils in the most deeply dividing branches of the phylogenetic tree. So it's evident that biofilm formation is an ancient and integral component of the prokaryotic life cycle, and hence it can be termed as a very key or important factor for survival in certain diverse environments. And recent advances show that biofilms are very structurally complex dynamic systems with attributes of both primordial multicellular organisms and multifaceted ecosystems. And this biofilm formation represents a protected mode of growth that allows cells to survive in hostile environments and also disperse to colonize new niches. And the implications of these survival and propagative mechanisms in the context of both the natural environment and infectious disease is what is being studied till date. And intriguingly, the visual characteristics of biofilms growing in diverse environments are very strikingly similar, indicating that there are very important convergent survival strategies that are conferred in part by structural specialization. And it is widely accepted that biofilm formation can provide a sort of protection to the cells from hostile environment conditions such as UV radiation, then extreme pH, then high salinity, then extreme temperature, high pressure, then tolerance to desiccation, drying up basically. And in addition biofilms can have an enhanced resistance or basically tolerance to antibiotics and other antimicrobial agents as compared to the planktonic form of planktonia or the freely floating form of these bacteria. So biofilms can therefore serve as a defense against antimicrobials, substantially quenching the activity of antimicrobial substances that diffuse through the biofilm in a form of inhibition identified as diffusion reaction inhibition, which can involve chelation by the complex formation or degradation of or enzymatic degradation of the antimicrobials. So in addition, slow growth within sections of the biofilm and the generation of dormant persistent cells further increase the survival of these cells to antibiotic actions. So therefore, biofilms are not only a shield against external insults, but they can also recognize and respond effectively to environmental stresses. In general, the time scales associated with different abiotic and biotic stresses can vary considerably. The stresses that occur on intermediate time scales usually cause transient regulatory responses mediated by genetic regulation mechanisms. So in Listeria species biofilm, Listeria monocytogenes, I think in Listeria monocytogenes, a type of biofilm in this bacteria biofilm formation, there is a considerable modulation of the protein abundance. It is observed as a function of temperature. And some scientists have observed and examined how different stresses induce profound differences in the biofilm basically physiology in terms of stratification of the matrix, microscopic architecture, then biophysical properties, and directly visible morphology of the macrocolony biofilms. And long-term exposure to any kind of constant stresses such as extreme environment, extreme pH, high salinity, all this can result in a process of mutation selection that results in a progeny with new genetic characteristics, and the biofilm features allow then a rapid and efficient adaptation to this long-term stress. So indeed, the high cell density that characterizes the biofilm, there's a very high cell density in a biofilm, right? So this high cell density and accumulation of mobile genetic elements within the matrix, it offers an ideal set of factors for efficient horizontal gene transfer. In addition, the matrix also provides a very stable physical environment for cell-to-cell contact, ideal for plasmid conjugation. Indeed, conjugation has been demonstrated to be much more effective and efficient in biofilms compared to the free-living bacteria in the plankton stage. And in biofilms, the accumulation of the genomic changes allow microbial evolutionary processes to occur in a very short time. And this creates new phenotypes that have advantages within the biofilm environment. And this has been reported, I think, in the case of evolution of Pseudomonas aeruginosa, which was associated with increased persistence in chronic cystic fibrosis infections, I believe. So these biofilms provide a very relatively stable and protected microenvironment for the cells, where they can change and adapt to the environmental conditions easily via transient changes in gene expressions as well as through permanent changes in the genome, and are fixed in populations but under natural selection. And these features make biofilms unique and central to microbial evolution and adaptations. At high temperatures, biofilms can withstand to the external high temperature and make the inside adapt for growth and reproduction. On the other hand, biofilm can also stabilize the inner environment when the outside is extremely cold, causing no freezing of the cells and enabling them to survive. So several scientific reports have shown this, that the biofilm mode of life stimulates the occurrence of genotypic and phenotypic variants. And such high diversity has an inherent risk, as a fraction of the new individuals could not be able to survive in a given environmental conditions. So in this situation, the supply of a productive environment like biofilms offers a huge advantage by allowing the new generation to be protected from the external selection conditions while continuing to undergo evolutionary process. So that's how biofilm provides a protective environment in which the new individuals with reduced fitness to external stress or the environmental changes can collect mutations that balance any kind of negative genetic changes. And in this way, the biofilm helps the microorganisms to adapt and evolve. Furthermore, the ability to evolve, the ability to create of biofilms to create gradients of selection conditions from the maximum environmental impact on the surface towards the minimum impact in the internal part of the biofilm gives the cells a unique opportunity to adapt before being exposed to the outside environment by the process of biofilm dispersion. So this process is markedly different from what occurs in other systems, where the diversity is fixed without the possibility of changes and further selection before regeneration. So this role of biofilms as diversity incubators, where the dynamic process of generating genotypic and phenotypic variants occurs, makes it the most successful strategy to colonize extreme environments and probably the only possible one. So I think that tells you how important, like how advantageous biofilm formation is for the bacteria. So this is another sketch to show you the importance. This diagram is not really well, but I'll read through it. Starvation can induce bacteria to shrink and adopt a spore-like state known as ultramicrobacteria, which wait in water, soil or rock or tissue until conditions suitable for active growth. Active bacteria can attach to any surface. Changes in gene expression transform swimmers to stickers within minutes, from plankton stage to the sessile stage within minutes. Attached bacteria multiply and encase the colonies with the slimy matrix. So there is a slimy matrix formation that happens in biofilm. Then the nutrients diffuse into the matrix. The close proximity of cells in the matrix facilitates the exchange of molecular signals that regulates the behavior. Core of sensing. Chemical gradients create microenvironments for different microbial species or the levels of activity. And then although antimicrobials damage outer layers, as I just said, the biofilm community is resistant. When the bacteria is present in the format of a biofilm, it's very difficult for antimicrobials or antibactericidals to show their effect. Propelled by shear forces, aggregated cells can become detached or roll or ripple along a surface in sheets and remain in their protected biofilm state. So these are some of the advantages of showing up in a biofilm state. Moving on to the next question. Higher the BOD, higher are the chances of the water to be contaminated with contaminants such as sewage. What do you think? I have talked about BOD, I think, in all the classes. BOD is biological oxygen demand. So anybody who would like to try, higher the BOD, higher the chances of water to be contaminated with contaminants. What do you think? It's true or false? Anybody?
True.
Correct. So I'll talk a little bit again about biological oxygen demand. So it basically measures the amount of oxygen consumed by microorganisms for the process of decomposition of the organic matters in any kind of water body. Basically, it indicates the amount of organic pollution present in an aquatic ecosystem, and it is calculated in sewage treatment or wastewater treatment to find the destruction of organic waste by aerobic microbes. It determines the amount of organic matter present in soil, sewages, sediment, garbage, sludge, etc. And this determines the rate of respiration in living things. So it's also BOD. This term is also very prominently used in any kind of medicinal and pharmaceutical industries to test the oxygen concentration of cell cultures. So as we know, drinking water, which is what we intake in our bodies, it has to be purer than garbage water, right? So drinking water has a BOD level of 1 to 2 ppm. So the demand for oxygen is really small. That is, the organisms using up oxygen, the quantity, the number of such organisms is really small. When the BOD value of water is in the range of 3 to 5 ppm, the water is moderately clean. Polluted water has a BOD value in the range of 6 to 9 ppm. In polluted water, some organic waste is present. That's why the polluted water, some organic waste, is a lot of microorganisms that take up a lot of oxygen for their action, and that's why a huge biological oxygen demand is created, and that's why the biological oxygen demand value in polluted water is 6 to 9 ppm, which is indicates very effective pollution. So this is a higher BOD or higher biochemical oxygen demand indicates that more oxygen is required, which is less for oxygen demanding species to feed on and signifies a lower water quality. Inversely, on the contrary, low BOD means less oxygen is being removed from water, so water is generally purer. This is what I explained. So unpolluted rivers usually have BOD levels below 1 part per million, 1 ppm, while untreated sewage has between 200 to 600 ppm, which is huge. You can compare yourself. Unpolluted rivers having 1 ppm and untreated sewage having 200 to 600 ppm. Next question. The purpose of disinfection is to kill all the microbes. Anybody? True or false? What do we disinfect any surface or anything for? What is the main goal of disinfection? Anybody just try? Okay. See, yes, it's true. The purpose of disinfection is to kill all the microbes, but disinfection kills most of the microbes, but there are some spores that go away scot-free even in case of disinfection. So let's talk about what disinfection is and what sterilization is. So as I said, disinfection describes a process that eliminates most of the pathogenic organisms except bacterial spores on any kind of inanimate objects. So in healthcare settings, objects usually are disinfected by liquid chemicals or wet pasteurization, and each of the various factors that affect the efficacy of disinfection can nullify or limit the efficacy of the process. So factors that affect the efficacy of both disinfection and sterilization include prior cleaning of the object, organic and inorganic load present, type and level of microbial contamination, concentration of and exposure time to the germicide, physical nature of the object, presence of biofilms, temperature and pH of the disinfection process, and in some cases relative humidity of the sterilization process. So the main goal is to kill all the microbes, but still there are some bacterial spores that are not wiped out completely in case of disinfection. But what is sterilization? Sterilization basically describes the destruction or the elimination of all forms of microbial life, and it is also carried out in healthcare facilities by physical or chemical methods. Steam under pressure, like how we do in autoclave, dry heat, ethanol ETO gas, hydrogen peroxide gas plasma cleaning, and liquid chemicals are the principal sterilizing agents used in healthcare facilities. And basically it is intended to convey an absolute meaning. Unfortunately, some technicians, some people use the words disinfection and sterilization interchangeably, but they don't exactly mean the same. So when chemicals are used to destroy all forms of microbial life, they can be called chemical sterilants. These same germicides, when used for shorter exposure periods, can also be part of the disinfection process. So unlike sterilization, disinfection is not sporicidal. Cidal is like killing. So it does not kill the spores, bacterial spores exactly. A few disinfectants will kill the spores with prolonged exposure time, like almost 3 to 12 hours. If you keep that exposure on, then disinfection might kill some of the bacterial spores, and these are called chemical sterilants at similar concentrations but with shorter exposure time, suppose 20 minutes and all that. These disinfectants will kill all microorganisms except large numbers of bacterial spores. So they are called high level disinfectants. Low level disinfectants can kill most vegetative bacteria, some fungi and some viruses in a practical period of time, like less than 10 minutes. An intermediate level of disinfectants might be might be killing the mycobacteria, vegetative bacteria, most viruses and most fungi, but they do not definitely kill the bacterial spores. And germicides differ markedly, primarily in the antimicrobial spectrum and rapidity of action. So these are the differences between cleaner, sanitizers, disinfectants, virucides and sterilants. So cleaner helps in soil removal. Cleaner simply adds in removing soil from a surface. Although cleaning does remove germs from surfaces, it does not kill them. Obviously it is not cleaning them. It's not killing them, it is cleaning the germs away from the surface. Sanitization reduces the number of bacteria. We have been using sanitizers in the last 2-3 years a lot due to the pandemic and all things. So sanitizer, basically they lower the number of bacteria on surfaces to levels that are considered safe to be used by public health organization. And these products tend to be faster and safer than disinfections because you cannot really sterilize our own body or pour spray disinfectants on our body. That's harmful for us as well. Sanitizers are a mild way to reduce the number of bacteria, so that's why we use sanitizers in our hands and we just pour a drop of sanitizer and we wipe our hands. Even prolonged exposure to these kind of sanitizers, like if you're using a particular sanitizer very frequently, which was the case during the COVID pandemic, people were actually very anxious, they were hysterical, they always wanted to use sanitizer because they were going out, which is understandable, and they were using sanitizer a lot. So at that time also, if you had noticed, doctors or any science professionals, public health professionals, they were preferring soap, washing your hands with soap more than sanitizer. So when you're washing your hands with soap, even when you're eating something, it's always preferred that you wash your hand with soap and not exactly with sanitizer. If you don't have any, for soap you need proper water supply. So if you are in a place where you are not able to access water, at that point during that time, sanitizer is fine. But using sanitizer all the time before having food and stuff is not quite right, because sanitizer basically reduces the number of bacteria because you're only exposing it to sanitizer for a short time period. Ready sanitizer, you're wiping it off. It's like for a few seconds you're exposing your hands to the sanitizer, and since the sanitizer contains a lot of content, I think 70% or 70% ethanol alcohol, it also vaporizes, so you're not exposing your hands to the liquid for a long amount of time. Soaps are much better. You are basically washing your hands for 20 seconds to 30 seconds. You are basically stripping off any kind of microorganisms from your hands, and also your hands are much more like soap is more pleasant to your hands than sanitizer. So that's why using sanitizer all the time was not really recommended. If you have easy access to soap and water, that should be the number one choice to clean your hands. Sanitizer in dire cases, when you don't have any kind of water access, you're on a trip or you're in a desert or somewhere and you don't have sanitizer, that's when soap, water, that's when sanitizer usage is fine. Disinfectant, obviously you cannot spray disinfectant on your body. I think if you have seen, we used to buy Savlon disinfectants, and on the bottle itself it's written that 99.9% killing, it performs the killing. You cannot spray all those disinfectants on your hands or on your skin, your hands, and all your body parts. The skin is very soft and it'll basically rot away if you use disinfectants like that. Those kind of disinfectants are not suitable for your skin. So, and you also expose so those are more harsh, so those are more, the chances are higher for those kind of disinfectants to be cleaning or killing the bacteria. Also, as I discussed, there are various types of disinfectants depending on the chemical composition and also the exposure time. Then you have virucide. Virucide kills viruses. It basically destroys or irreversibly inactivates viruses in any kind of inanimate environment. Then there is sterilant. Sterilant eliminates all fungi, bacteria, viruses and spores, and some kind of sterilants, chemical sterilants, are also used in the disinfection process, but for a shorter time span. Sterilization mostly happens with steam, water, like high pressure water and steam, all that kind of the concept of autoclave comes from this sterilization technique. So these are some of the differences between the various terms that we use in terms of killing microbes. So this is another difference between disinfection and sterilization. So disinfection is the process of elimination of pathogenic microorganisms. You see how you generally wear a glove while we're using these kind of things, because disinfectants can be really harsh to your skin. However, the process is not effective in the case of vegetative spores. But sterilization refers to any process that eliminates, removes, kills or deactivates all forms of life in biological agents present in a specified region. So you can see in biology labs, you will know that there is an instrument called the autoclave, and in all biology labs it's an indispensable part of biology lab. Autoclaves are used to sterilize any kind of container, any kind of plates or whatever, the media, whatever you're supposed to use in the culture process afterwards, so as to remove traces of any previously grown organisms over there. Yeah. So moving on to the next question. Piezophilic microbes survive under high pressure. This is also a similar kind of question we discussed in the last session also. Anybody? Piezophilic, just tell me what's the meaning of piezophilic. Piezophilic microbes survive under high pressures. If you know the meaning of piezophilic, that will be, then you know the answer. Anybody? Piezophilic microbes. I'm still in the meeting, right? Yeah. Anybody?
Organisms that lives under elevated hydrostatic pressure.
Yeah. So philic means to love. Anything hydrophilic, lipophilic. Philic means to love, and phobic is like phobia. Phobia is a very common term that we use when we are scared of something. So that's phobia. So philia and phobia are basically opposite terms. So when we say piezophilic, that means organisms loving high pressure. So we know that there's, when we talk about the ocean has an average depth of 4,000 meter almost, and therefore there will be a pressure of almost 40 megapascal, and one, like 0.1 megapascal is 1 bar. So we can understand how much pressure that might be. So at the bottom of the Mariana Trench, the deepest known site in the ocean, the pressure is 110 megapascal. So despite the fact that a major proportion of the Earth's biosphere is high pressure environment because of the oceans, we have a major portion of the Earth is ocean and seas. So despite this fact that the major proportion of the Earth's atmosphere is the biosphere is the high pressure environment, much less is known about these deep sea piezophilic or barophilic. It's also called barophilic microorganisms. The temperature in the deep sea is typically within the range of 1 to 3 degrees Celsius, and the discovery of certain hydrothermal vents in the seafloor has revealed habitats where high pressure and high temperature still conditions exist. And thus marine organisms basically inhabit environments where they might be exposed to temperature range of 1 to 300 degrees Celsius and pressures that range from 0.1 to 110 megapascals. So deep sea organisms thus have attracted a lot of attention from marine biologists who wish to understand the survival strategies employed under such extreme conditions. So yes, this is true. So piezophilic organisms possess optimal growth rate at pressures above the atmospheric pressure, and piezo-tolerant microorganisms are capable of growth at high pressure as well as at atmospheric pressure, but can be distinguished from the piezophiles because they do not have optimal growth rates at pressures above 1 atmosphere. So piezo-tolerant microbes can also be distinguished from piezo-sensitive microorganisms. These are the piezo-sensitive microorganisms whose growth is sensitive to the elevated pressure because they can grow at 50 megapascals at a rate that is above 30% of their growth rate at atmospheric pressure, as long as they have otherwise optimal growth conditions. So it is important to know that various physiochemical properties or conditions can have a large influence on the effect of these high pressure and microbial growth rates. So yes, piezophilic microbes survive under high pressure. That is very true. They love surviving under high hydrostatic pressures. Which of these was a breakthrough principle of Sanger's sequencing method? Anybody? Anybody would like to try?
Use of DNA synthesis.
Only that one. This is a multiple choice question. So there's circular, there's square boxes. Anything else that you would like to add? Which was a breakthrough principle of this technique? This is also called the chain termination technique. So what did they use that helped them terminate the chains of DNA synthesis?
Use of DDNTP.
Correct. DDNTP is dideoxy nucleotide triphosphates. So these are the two options that are correct for the Sanger sequencing, but we'll also explain a little bit about what Sanger sequencing is. So it is also known as the chain termination method, and this was developed by this biochemist Frederick Sanger and his colleagues in 1977, and he got a Nobel Prize for this, I think in 1980 or something. I'm not sure about the year, but this person got 2 Nobel Prizes. One prize he got in the 1950s and that was because of his research on insulin, and then he got another Nobel Prize for this thing. So this method is designed for determining the sequence of nucleotide bases in a piece of DNA. And so when you get an unknown DNA piece, you don't have any idea of the sequence. So this is basically what was used, one of the first sequencing techniques to basically recognize the nucleotide sequence. And Sanger sequencing was formed with at least 99.9% base accuracy and is still considered the gold standard for validating DNA sequencing, including those already sequenced for next generation sequencing. And Sanger sequencing was also used in the Human Genome Project to determine the sequences of relatively small fragments of human DNA, and these fragments were then used to assemble larger DNA fragments and eventually entire chromosomes. So even till this day, Sanger sequencing is a very important technique today. So it's a method that yields information about the identity and order of the 4 nucleotide bases. ATGC, we know that there are 4 nucleotide bases: adenine, thymine, guanine, cytosine, and how these the identity of these 4 nucleotide bases and the order of these 4 nucleotide bases is what we can get to know from Sanger sequencing, and this is also known as the chain termination method. And so what happens in the mid 1970s, Sanger was not really alone in this race to sequence DNA. So it's not, competition does not only exist in today, even at that point of time there was a huge competition in various discovering the various techniques, like how to do how to do sequencing basically. So almost in parallel, when this Frederick Sanger was working on this technique, almost in parallel, two American scientists, Maxim and Gilbert, were also trying to develop a technique. They developed a technique in which DNA is chemically treated to break the chain at different specific bases, and following electrophoresis, or following separation of these cleaved DNA, the relative lengths of the fragments and thus the positions of the specific nucleotides could be determined and the sequence inferred. So that was considered the birth of the first generation sequencing. However, the advent of this Sanger sequencing in 1977 would be the breakthrough that propelled sequencing into the future. Many years even after its development, Sanger sequencing was used to sequence the entire human genome. So what happens, Sanger sequencing, is that it targets a specific region of the template DNA using an oligonucleotide sequencing primer, which binds to the DNA adjacent to the region of interest. So there must be an area of known sequence close to the target DNA. And in order to determine the sequence, Sanger sequencing makes use of chemical analogs of the 4 nucleotides in DNA. So these analogs are called dideoxyribonucleotides, or DDNTPs, and they basically are missing the OH, the 3' hydroxyl group that is required for the 5' to 3' extension of the DNA polynucleotide chain. So if this is the normal base, this has this OH. So this OH will further initiate the formation of this chain. This OH will help in the 5' to 3' extension. Another DNTP will come and attach to it, then another will attach to it. But if in place of OH we have H, then there's no free space, there's no site for it to attach to another nucleotide base. Basically that will terminate the chain. No more 5' to 3' extension will happen, and that's why this is called the dideoxy, because the OH is replaced by H. It's a chemical analog, but the OH is replaced by H, and if this dideoxynucleotide is incorporated into any chain, then the chain will get terminated. After this incorporation, no further nucleotides will be able to add to it because of the absence of the OH group. So by mixing different DDNTPs that have been labeled with different color for each base, unlabeled DNTPs, and a template DNA in a polymerase driven reaction, strands of each possible length were produced. When the DDNTPs are randomly incorporated, and these terminate the chain, and the extension products are then separated by electrophoresis, that is resolved to single nucleotide differences in size, and the chain terminated fragments are detected by their fluorescent labels, with each color identifying one of the terminated DDNTPs. The sequences of the DNA strand can thus be derived from analysis. So this is how the DNA sequencing by Sanger method work. The polymerase, there will be a polymerase enzyme which will extend the primer by adding the complementary DNTP to the template DNA strand. So to determine which nucleotide is incorporated, so you have the strand in which the DNA sequence is unknown, and then you are trying to do polymerase chain reaction of that strand. So obviously complementary nucleotides will come and attach to the complementary strand. So to determine which nucleotide is incorporated into the chain of the nucleotides, the 4 dideoxy triphosphates, which were labeled with the fluorescent dye, were used to terminate the synthesis reaction. So once these dideoxynucleotides are added, the chain is terminated and it cannot form a link with the next nucleotide. And following the synthesis, the reaction products were then loaded into 4 lanes of the single gel, depending on the diverse chain terminating nucleotides, and then gel electrophoresis, which is basically a technique to separate DNA based on their molecular weight. And according to the sizes, the sequence of the DNA is thus determined. So even one single nucleotide base difference had to be identified from that gel. So you can imagine the kind of resolution that gel needed to have to understand. In today's time, we run agarose gel, we can understand the difference in the molecular weight. But to even recognize the difference in one base pair, the gel had to have a very high resolution, slow timing, huge gel. That were the main requirements. So these are the steps of the Sanger sequencing. It's a double-stranded DNA. It is basically this double-stranded DNA. We don't know the sequence of. We want to know the sequence of this DNA. So we denature it into two single-stranded DNA or ssDNA. Then a primer that corresponds to one end of the sequence. A primer, we have to know a little bit of the sequence from beforehand. So a primer that corresponds to one end of the sequence is attached. Then 4 polymerase solutions. Then there are 4 tubes that will have the 4 polymerase solutions with 4 types of DNTPs, but only one type of DDNTP. So these 4 tubes will have all the DNTPs like normal deoxyribonucleotides, but each tube will have only one dideoxy nucleotide. That means the purple tube will have dideoxy for the A nucleotide base. The yellow tube will have dideoxy variant for the T nucleotide base. The orange one will have dideoxy for the C, and the green one will have for D. So the DNA synthesis reaction is then initiated, and the complementary chains grow until the termination. So if there is an A, if on the template strand there is a T thymine, so that means an A should come in the complementary strand. So if A, that is the dideoxyadenine base, is attached, the chain will be terminated. Similarly, when there is cytosine in the template strand, GC, so G, this guanine should come in the complementary strand. So once the dideoxy of the guanine nucleotide comes and attaches to it, the chain is terminated. And so this chain DNA synthesis reaction happens in all the 4 tubes until the termination nucleotide comes and is randomly incorporated. And this resulting DNA fragments are then denatured into, then these are again denatured so as to separate the double strands, and then loaded into the electrophoresis chamber in the 4 different wells. And these denatured fragments are then separated by electrophoresis to even recognize even one single base pair difference, and then the sequence is determined. So as you can see, over T is over here, so there's T, then there's C. This is how the entire sequence of DNA can be figured out. So this will be the complementary strand. So we can obviously find out the complementary and the complementary, that means the actual sequence of the template strand. So this is how the Sanger sequencing worked, and that's why the first 2 options, use of DNA synthesis and use of DDNTP, were the breakthrough principles of this technique. Moving on to the next question. Which of these match best with their attributes? First generation sequencing, second generation sequencing, third and fourth, and these are the 4 options. Anybody? Anybody would like to try?
Option 2.
Option 2 is 1D, 2B, 3C. Correct. So we'll talk a little bit about the different generations of sequencing. So I have already talked about the Sanger sequencing. This is Frederick Sanger, as you can see. So Frederick Sanger was the one who brought in the breakthrough in the DNA sequencing, which is basically the chain termination method, and this method went on to dominate the sequencing world for the next 30 years, and that is why this person is considered as a giant in genomics. He was, as I said, he was awarded the Nobel Prize for his revolutionary work in 1980, but it was his second Nobel Prize. So his first Nobel Prize was in the 1950s, 1956 or 1958, I think, 1958, which was awarded for his work on insulin, on the structure of insulin. And so this is the entire chain termination technique. In 1977, Sanger also used this method to sequence the first ever complete genome the one of the bacteriophage 5X174. It's a bacteriophage that infects E. coli, and later it became one of the most popular DNA positive control in labs around the globe. So around in 1977 only, there were two American scientists, Maxim and Gilbert, who introduced a method for DNA sequencing that was based more on the chemical modification of DNA. So that method involved including chemicals that break the DNA sequence at specific bases, and in contrast to Sanger sequencing, it did not really rely on DNA polymerase. However, both Sanger sequencing and Maxim Gilbert sequencing lacked automation and were very tedious, tiring, and time consuming. So nevertheless, the research community had recognized the potential of this Sanger sequencing by them, and many research groups worked on the automation of this process. So by then there were more techniques that were coming up, and so this is the second generation sequencing, where luciferase is basically used, and you can see that this is a template. This is a new sequence. There will be a polymerase which will show an action. There will be a luciferase that will prompt the polymerase to show different light peak at a particular wavelength. There will be DNTP coming in, and depending on that there will be light peaks shown on the graph. So then there were ion semiconductor. So then there was the third generation DNA sequencing, where basically nanoholes then required the third generation came from the second generation, where there were single molecule real-time sequencing technologies that were defined as the third generation, and there were packed biosequences that were happening. Then ultimately there was the next generation sequencing that took the floor, which included various technologies that perform sequencing and gather data from multiple reactions running simultaneously, and it was also known as the massive parallel sequencing. And like next generation sequencing basically followed the Sanger sequencing technique only, but however in Sanger sequencing there were separate steps for sequencing, separation and detection, whereas in next generation sequencing it basically uses an array based technology to do everything simultaneously. And even though there are many next generation sequencing available, all of them basically follow the three basic steps. There will be a sample or library preparation. A library will be prepared by fragmenting the DNA sample and ligating it with commercially available adapter molecules, and adapter molecules will act in the hybridization of the library fragments to the matrix, and adapter molecules will provide a priming site. Then there will be amplification and sequencing. The library will be converted into single stranded molecules, and amplification will finally create clusters of DNA molecules, and each cluster will act as an individual reaction where sequencing is performed. Then there will be data output and analysis, the final step. At the end of the reaction, each NGS run will provide a large amount of raw data, and this data can be used to sequence whatever is happening. So, and the final, that is today we are talking about the nanopore sequencing. So if we talk about the nanopore sequencing, that is the most portable, and in today's time it is the most easy to do technique. So nanopore based sequences, these are the fourth generation DNA sequencing technology, and they can actually very quickly sequence the entire human genome for less than $1,000. The single molecule techniques used by this technology allow us to further study the interaction between DNA and different proteins, as well as between protein and protein. So nanopore analysis opens a new door to molecular biology investigation at the single molecule cell. So DNA, as we know, it's a molecule that encodes genetic instructions, a blueprint of life. So basically accurate and rapid DNA sequencing will have profound impacts on human disease and personalized medicine. So nanopore is nothing but a small hole on the order of I think 1 nanometer in internal diameter that is made up of certain transmembrane cellular proteins. And nanopore sequencing works on the principle of minute changes in the electric current across the nanopore immersed in a conducting fluid with the voltage applied when a moving nucleotide from a DNA strand passes through it. So every nucleotide on the DNA molecule while passing through the nanopore will obstruct the nanopore to a different characteristic degree, and the amount of change in the current is characteristic for each different nucleotide, and DNA may be forced to pass through the whole one base at a time, like thread through the eye of a needle. And the change in the current can be directly read, and the sequence of the passing DNA can be determined by detecting changes in the current generated specific to the base passed. And alternatively, a specific nanopore can be designed to produce current changes when a specific nucleotide passes through it. So although this technique has not been in use for sequencing in a routine manner, according to the scientists who developed it, it can actually sequence the entire 5.4 kilobase genome of the virus in one continuous read. However, it initially aims to deliver reads of I think 1000 kilobases. That is still much longer than the fragments processed by any other technology. And the advantage of this nanopore system is that it could deliver real-time sequencing of single molecules at very low cost. It is also expected to read very long DNA molecules in a single read. And nanopore technology is expected to provide sequencing at a very low cost, like $40 or $50 per gigabase of sequence. That would mean a cost of just a few $1,000s to sequence a human genome to the sequence of to the standard of 30-fold coverage. So it's all not only is it a very fast sequencing process, it could actually be the future choice of sequencing for nearly all applications. However, the limitations are that this is still in the very proof of concept stage and not yet commercially available. At this stage, there are certain concerns about this technology that are still yet to be taken into account, and one of these is to be how to improve its resolution so that it can detect single bases with the rapid movement of DNA through the hole. So this is the nanopore sequencing. Obviously, this is very portable. The instruments are very small and easily portable. Moving on to the next question. Choose the correct option. Altered current is measured to identify the passing base pair in nanopore sequencing. Anomalies in codon bias can be caused by horizontal gene transfer. Which one is true? Anybody? Okay, so both 1 and 2 will be true. Altered current is measured to identify the passing base pair is what I already discussed. So there will be different alteration in the current in the nanohole depending on the different bases that are passing when the DNA structure is passing through the nanopore. So that is exactly what happens in case of nanopore sequencing. Anomalies in the codon bias. So when horizontal gene transfer is happening, mostly three categories: conjugation, there's conjugation happening, there's transformation, there's transduction. These are the three most widely described techniques for horizontal gene transfer. So when the gene is getting transferred from one organism to the another, there will be obviously differences in the codon bias, because the host organism, the source organism will have a different codon bias than the recipient organism. So I think we know what a codon means. It's a set of 3 nucleotides that encodes a particular amino acid, and one particular amino acid can actually be read by many different codon sequences. Mostly the third codon is the one that is changed the most, and that is why the third position is called the wobble sequence. So as you can talk about the translation, so transferred gene with efficient codons. So one particular gene that has very efficient codons for recognition by that particular organism's ribosome is being transferred to another organism, and transferred gene with non-efficient codons. So the other organism the same codons might not be that kind of efficient for ribosome recognition in the recipient organism. So that is what is being mentioned over here. So this diagram is showing the relation between translation efficiency and the horizontal gene transfer. So transferred genes with neutral function but with codons that are adapted to the host. So these codons are very easily recognized by the host ribosome, and the host ribosome knows which amino acid these codons signify. These have a smaller effect on the fitness of the host as they recruit less ribosomes than genes with codons that are not adapted to the host. So if there are codons that are not properly adapted to the host only, that will have greater effect on the fitness of the host. And then B shows a duplication or horizontal transfer of a tRNA that recognizes rare codons in the transferred genes may improve the translation efficiency of these genes and thus decrease the ribosomal density. So if the tRNA is also horizontally transferred, and if this tRNA is capable of recognizing rare codons in the recipient organism, that might actually help improve the translation efficiency of these genes and then decrease the ribosomal density. So after such a change in the tRNA pool, the number of available ribosome increases and thus the fitness of the host is augmented. So that is what in any kind of anomalies in the codon bias can be very easily caused by horizontal gene transfer. Moving on to the next question, which of these is false? BLAST P or protein BLAST gives a more reliable picture of functional similarities than BLAST N or nucleotide BLAST. Green genes is the dataset used for BLAST algorithms. Transcriptomics is highly time sensitive, and microarray is based on the principle of DNA RNA hybridization. What do you think? Anybody? Anybody would like to try? I'll wait for a few minutes. You can read, this is a multiple choice answer, so there can be more than one correct answer.
Option B.
Option B is false. Anything else? Yes, option B is false. Option D is also not exactly correct. So I'll explain all of these a little bit. So first the option was about BLAST. So BLAST is obviously a program that is used online. You can just go on Google and search BLAST and BLAST P. So you can actually put two FASTA sequences of nucleotide or two protein sequences, and they can actually compare the number of hits that each portion of the sequence will have with the other portion. So BLAST N means when two nucleotide sequences are compared or try to compare against each other. BLAST P is comparing protein with protein. BLAST X is comparing translated nucleotide with protein. And BLAST N is translated protein with nucleotide, and BLAST X is translated nucleotide with translated protein. So these are different BLAST programs. You can just go on Google, search it, and you can actually put two FASTA sequences of any two nucleotides to any two proteins or anything, and you can actually compare the number of hits that these things are getting. So yes, so BLAST P gives a more reliable picture of functional similarities, because if we are looking for a certain protein encoding, so this statement is false. That means BLAST P does not give BLAST P this statement is correct, because BLAST P gives a much more reliable picture of functional similarities than BLAST N, because if you are looking for a certain protein coding gene of interest in the genome of some random organism, you often may have the choice of running either the BLAST N or the BLAST P program. In the former case, like in case of running the BLAST nucleotide program, you would use the gene of interest mRNA sequence as a query and the genome of the organism as a sequence database. So if you have the mRNA sequence, you will put that and you will try to see if it is forming it is having any kind of hits with the genome of that particular organism. But if you are using BLAST P or the protein BLAST, you have the protein sequence of the gene of interest as a query, and you will be comparing that against a database comprising the proteome of the entire organism. So you may think the results of these two searches should be comparable or identical. So after all, if the organism in question has a homologue of the gene of interest, in both the nucleotide and the amino acid sequence should be identified. But in fact, BLAST protein found a homologue of the human AGO2 sequence with an e-value of 3 × 10 to the power minus 38 in the proteome of a single-celled ancestor of animals. So when repeating this search using the AGO2 mRNA sequence as a query and the genome of that particular bacteria as a database, no hits were reported at all. So what is happening here? Why are BLAST P and BLAST N showing different kinds of results. So BLAST nucleotide is actually a very poor tool for finding protein coding sequences. This is in part due to the global position of the third nucleotide in most codons, as I talked about in the last question also. The last position of nucleotide in every codon is the global position. So that changes, so that can change, and even then that particular codon might encode for a particular amino acid. So a particular amino acid can be coded by at least 4-5 codons, can be coded by 4-5 codons also, only the last vowel position changing. So the exact amino acid sequence can be encoded by 2 nucleotide sequences differing in every third position. Since mutations in the third position do not affect the resulting protein, such mutations typically accumulate quite rapidly. And the amino acid sequences being identical, BLAST protein would have no problem in retrieving one sequence using the other sequences query. BLAST nucleotide, however, uses a default word size of 11 nucleotides. This means that the two sequences must match with at least 11 nucleotides for BLAST nucleotide to be able to report any hit at all. So in the above example, like if you're talking about this, a perfect match in case of BLAST nucleotide is actually it does not provide alignment very properly. So that's why BLAST nucleotide is very poor tool to search for functional similarities. The obvious solution is to use BLAST protein rather than BLAST nucleotide whenever possible. So that's how BLAST nucleotide is not really correct. So that the first option is correct. So for this question, since the question asks which of these is false, the first option will not be in the answer because it's correct. Green Genes is the database used for BLAST algorithms. Green Genes is actually a full-length 16S rRNA gene database that provides a curated taxonomy based on the de novo tree inference. So it's a 16S rRNA gene database, so it's false. Transcriptomics is highly time sensitive. Definitely transcriptomics is very highly time sensitive. So these are the different omics approaches that are generally taken on in biology labs. Genomics basically talk about the entire genome of an organism. Then transcriptomics talks about the transcriptome or the RNA level of the organism, and the translation, after translation proteins are formed, and the proteins or protein arrays talk about the entire proteome of the organism. So transcriptomics, since they talk about mRNA, and mRNA basically is very easily degradable, like DNA is very stable but mRNA is not, it can degrade very easily. That's why transcriptomics is very time sensitive. So in case if you want to analyze the transcriptome of any organism, you should if you do the analysis at 2 different time points, I'm pretty sure there'll be very huge differences in the results. So that's why transcriptomics is very time sensitive. Moving on to the last once microarray is based on the principle of DNA RNA hybridization. So microarray technology is basically a laboratory approach that involves binding an array of thousands to millions of non-nucleic acid fragments to a solid surface referred to as the chip. And the chip is then bathed with DNA or RNA isolated from a study sample, such as cells or tissues, and then complementary base pairing between the sample and the chip immobilized fragments will produce light through fluorescence that can be detected using a specialized machine. So microarray technology can be used for a variety of purposes in research and clinical studies, such as measuring gene expression and detecting specific DNA sequences. So mRNA is an intermediary molecule which carries the genetic information of the cell nucleus to the cytoplasm for protein synthesis. So whenever some genes are expressed or are in their active state, many copies of the mRNA corresponding to the particular genes are produced by a process called transcription, and these mRNAs synthesize the corresponding protein by translation. So indirectly, by assessing the various mRNAs, we can assess the genetic information of the gene expression, and this helps in understanding of various processes behind every altered genetic expression. So mRNA acts as a surrogate marker basically. But since mRNA is degraded so very easily, it's not stable at all, it is very necessary to convert it to a more stable cDNA form. cDNA is the complementary DNA form. mRNA can be reverse transcribed to the cDNA. So, and then labeling of this complementary DNA or cDNA is done by fluorochrome dyes such as Psi3, which is green in color, or Psi5, which is red in color. And the principle behind microarrays is that complementary sequences will bind to each other. So what happens is that the unknown DNA molecules are cut into fragments by restriction enzymes. In the nucleuses, the fluorescent markers are attached to these DNA fragments, and these are then allowed to react with probes of the DNA chip. Then the target DNA fragments along with the complementary sequences bind to the DNA probes, and the remaining DNA fragments which are unbound or loosely bound are washed away. The target DNA pieces can be identified by the fluorescence emission by passing a laser beam, and a computer is then used to record the pattern of fluorescence emission and DNA identification. So this technique of employing DNA chips is very rapid, besides being very sensitive and specific for the identification of several DNA fragments simultaneously. So the study of the expression of most genes in a specimen is not hypothesis driven, and most of the studies refer to it, but instead it's kind of can be figured out through this microarray technique. So DNA microarrays are basically solid supports, usually of glass or silicon, upon which DNA is attached in an organized predetermined grid fashion. Each spot of DNA is called a probe, and it represents a single gene. And DNA microarrays can analyze the expression of tens of thousands of genes simultaneously. And there are several synonyms of DNA microarrays such as DNA chips, gene chips, DNA arrays, gene arrays, biochips, etc. So this is the basic principle of DNA microarrays. It basically lies on the hybridization between the nucleic acid strands, and the property of the complementary nucleic acid is to specifically pair with each other by forming hydrogen bonds between complementary nucleotide base pairs. And for this samples have to be expressing fluorescent dyes. So as you can see from the sketch over here also, when it's black, that means there is no fluorescent activity happening. So only the DNA that was the probe DNA that was attached to the chip is there. No DNA from the sample is attaching to it. Everything has been, nothing has attached properly to it. So there's not this particular primer, whatever this particular DNA chip was referring to, whichever gene this chip was referring to is not in that particular cell. So that is why it is not present in cell. If it is present in both the cells, it will fluoresce in both green and red. Suppose there is gene A that is present in both the normal cell and also the cancer cell, and the gene A's complementary strand has been attached here. So when we wash the entire chip with both the samples, both gene A from normal cell and both gene B from the cancer cell will attach. But gene A from normal cell is green in color, and gene B from gene A from cancer cell is red in color. So when both will attach, it will give the yellow color. That is why yellow means present in both the cells, because both the colors are showing, and simultaneously we can see the yellow color, not only green or red. But only if green is showing, that means this particular gene is only present in the normal cell and nothing is present in the cancer cell. That's why only green color is showing, because normal cells genes are fluorescently labeled with the green fluorescent probes. If it is showing only red color, that means this particular gene is present only in the cancer cells. So two samples are simultaneously hybridized to the chip, and complementary nucleic acid sequences between the sample and the probe attached to the chip get paired by the hydrogen bonds, and these non-specific bonding sequences while remain unattached and washed out during the washing step of the process. So then fluorescently labeled target sequences will bind to the probe sequence and will generate a signal. It can be no signal, or black, yellow, red or green. And the signal depends on the hybridization conditions, washing after hybridization, etc. And the total strength of the signal will depend upon the amount of the target sample present. So if you can see a very, if you are comparing two red signals, if you see that one signal is very bright and the other is not that bright, that will mean this particular gene is very heavily expressed in the cancer cell. So, and the other gene is also expressed in the cancer cell, that is why it's showing red in color. But if it is not showing that much intensity, that means not a lot of DNA has attached to it. So the intensity will actually talk about the total strength of the signal, which will further talk about the presence of that particular gene in the samples. and using this technology, the presence of one genomic or cDNA sequence in one like 1 lakh or more sequences can be easily screened in a single hybridization. So that is what the principle of DNA microarray technique is all about. And DNA microarray technique is basically talks about DNA hybridization, because mRNA is not at all stable. So that's why mRNA is converted to the cDNA or the complementary DNA form, which is by the process of reverse transcription, and then DNA hybridization takes place. So that is how this occurs. So we'll stop here today because it's already 8. So does anybody have any questions regarding to the DNA microarray technique or anything? We'll start the next session from the next day, next Friday. So I'll upload all these slides today only, and I'll upload the recording of this session also. If you have any questions later on also, I can take those in the next session. So if you don't have any questions today, maybe we can stop the session for today, and we can conclude the session. Anybody? Okay. Thank you so much for staying for 2 hours on a Friday night, and thank you for interacting as well. We'll continue with the questions from next Friday. Thank you.